Hi guys, hope you all are well. Welcome to my channel Shrutapa's Biology. I am Shrutapa Datta. Now I am continuing the topic biomolecule. In my last video, I have told you about the different nature and functions of the primary metabolites like that carbohydrate, protein and fat, about their the molecular structure and linkage etc. Bonds etc. About that. Now in this video, I shall give you the a framework or the basic concept about the nucleic acids, because nucleic acid is also considered as the macromolecule. In biomolecule, in my last video, you have seen that two types are there. One is micromolecule and another is macromolecule. Now in macromolecule, which have more than 1,000 molecular weight, in them nucleic acids are also included. So now I am going to that topic, that is the nucleic acids. So biomolecule 2. And the main topic of this part is nucleic acid. We know that the two types of nucleic acids are important. Number one is DNA or deoxyribonucleic acid. Both these nucleic acids are the genetic material. Number one is DNA or deoxyribonucleic acid and number two is RNA or only ribonucleic acid. These two names are popular and you know, I think from lower classes, and these two are the genetic material. सबसे बड़ा बात है ये genetic material है, तो इसीलिए इतना important है ये. In mostly higher organisms, we can get that the DNA is the genetic material, and in case of lower organisms, RNA is the genetic material generally. But in higher organisms, sometimes RNA has the role in the genetic, role in the genetics. Now, DNA has, about the detailed structure of DNA, if we study, then we shall get there are three parts. That is unit of the nucleic acid, or DNA is DNA, RNA both. The unit of nucleic acid, what is the unit of nucleic acid? Unit of nucleic acid is nucleotide. Now what are the components of the nucleotide? The basic components of the nucleotide is nitrogenous bases, N2 bases, ribose sugar. We need to say pentose sugar, as it has two kinds, ribose and deoxyribose. Pentose sugar, which has two divisions, ribose in RNA and deoxyribose in DNA, and another is phosphoric acid. So these are the three components of the nucleotide, and if anyone, that is phosphoric acid, is absent, nucleotide minus phosphoric acid acid equals to nucleotide. Sorry, nucleoside. Nucleotide I have told. This is nucleoside. So what's the difference between nucleoside and nucleotide? That the nucleoside, in case of the nucleoside, the phosphoric acid is absent, and in nucleotide, phosphoric acid, pentose sugar, and nitrogenous bases all are present. So nucleoside is incomplete nucleotide. Now about the nitrogenous bases. What are the nitrogenous bases? In DNA, we can get four kinds of nitrogenous bases. They are grouped into two divisions, that is purine and pyrimidine. N2 bases are purine plus pyrimidine. Now purine, what is purine? In purine, the components are adenine and guanine. Adenine plus guanine, and in case of pyrimidine, thymine plus cytosine in DNA, and in place of thymine, uracil and cytosine in RNA. In place of thymine, uracil is another type of pyrimidine which is present in RNA. So uracil is the nitrogen base which present in RNA, and thymine is present in DNA. Now this is about nitrogen bases, and then sugar is pentose sugar. I already have told you, that means 5 carbons are there to form the ring, and another is phosphoric acid. Now I am telling about the difference between purine and pyrimidine. The basic difference between purine and pyrimidine, that purine has 2 rings, 2 carbon rings. One is just like the pyrimidine, and another is extra ring is known as imidazole. So purine has 2 rings and pyrimidine has 1 ring. Pyrimidine has single carbon ring, and purine has two carbon rings, and the extra one is imidazole ring. Now about the DNA, I am telling that this is the main DNA is the main genetic material in different living organisms, and in our case, in human being also, DNA is the main genetic material. So DNA can be transmitted from one generation to other and can carry different characteristics. In our body, the DNA is the blueprint. Like for the construction of a building, for a construction of any civil work like the bridge, etc., the engineers, they need the blueprint. And here in human body, the DNA is the blueprint. That means all the plans of the human's body can be realized or understood by the geneticist by observing or studying their DNA. So DNA is the blueprint of body. DNA is the blueprint. Now DNA is present in all the cells. In each and every cell, in our body cell, in our germ cell, everywhere DNA is present. Now how can we study the DNA? About the study of DNA, firstly the Franklin and Wilkin, both the Wilkin and Franklin, both of them, Wilkins and Rosalind Franklin, okay, I am writing this Franklin, they performed the X-ray crystallography. What is X-ray crystallography? That is the study by using the X-ray diffraction method. Okay, so by X-ray crystallography, they performed and obtained the structure of DNA. On that basis, Watson and Crick, another two scientists or another two geneticists, we can say that the Watson and Crick, they have proposed a model of DNA, and Crick proposed the model of bDNA. bDNA. Now what is bDNA? bDNA is the most common and abundant DNA in living organism and human being also. bDNA is the most common one. And about the other DNAs, they are A, B, C, D. A, C, D. B is here. And other than B, there are A, C, D and Z. Another 4 types are also observed, and about their base pair content, about their coiling pattern, etc. They are different, and on the basis of that, these DNAs are characterized or categorized. Now you listen about this, that what is the bDNA and what it has the difference with the other type of DNAs. In case of the aDNA, the number of base pairs are different than the bDNA. The bDNA is the standard one and it's the most abundant and popular DNA, and bDNA has the base pair number 10. bDNA has 10 base pairs and its right-handed column. aDNA, it has more than 10 base pairs. It's also right-handed coil. More than 10, as I'm telling, but it's actually not the round numbers, that's why 10.58 or like this. And in case of cDNA, there are 11 base pairs. They are also right-handed coil. So you are thinking that then why I am telling in all cases if they are right-handed coil? No, the exception is, we shall get the exception. dDNA also right-handed coil, but zDNA has base pairs and its left-handed coil. Only one DNA which is left-handed column. Now in our body, more or less all the DNAs are present, but mostly the bDNA. And so about the structure of the bDNA, which is proposed by the Watson and Crick on the basis of the X-ray crystallography of Wilkins and Franklin. That's the standard description about the DNA. And in my next video, I shall continue about that. And I shall give the basic structure of the Watson-Crick model, which is very important for your 12 board class, 11 board class, and also the competitive exams. Very important. So just watch that. So bye for today.